Hello, my name is Isha Randawa, and for my science communication project, I wanted to focus on a topic that I found really interesting through researching several different articles where they were able to find that mushrooms are able to help us in repairing damaged nerves, and therefore they help in this process, they help support this process that we have going on developmentally in our nerves called neurogenesis. And this is of particular interest because of the fact that if we can use mushrooms in this way, we can actually uncover new alternative methods of treatment for neurodegenerative disorders. So here's some background on um, neurodegenerative disorders that are relevant to this presentation. Obviously, they are extremely complicated, and each of these disorders that I have listed here on the right definitely are deserving of their own presentations. But I found it necessary to just include some of the abbreviations that are very common so that you can also equip yourself in recognizing some of these disorders. Um, so, what are neurodegenerative disorders? Neurodegenerative disorders are specifically characterized by the intensifying death of nerve cells due to their condition. So, we have Parkinson's disorder, which is PD. Parkinson's disorder is a neurodegenerative disorder that is similar to Huntington's HD. It has a lot of motor function. It has a lot of effect on your motor function as well as your cognition. Alzheimer's, it's a pretty well known one, AD. Again, it's a neurodegenerative disorder specifically impairing your cognition. People obviously facing, like, it's very terrible seeing people going through or seeing your family members probably go through something like dementia. You really do get to see that, like, it is like a degeneration of their memory, of their capabilities of understanding the world around them, of their grasp on their sense of self. MS is another neurodegenerative disorder that has an effect on your motor function, so is motor neuron disease and prion disease. All of these are common neurodegenerative disorders. Now, what is neurogenesis? Neurons are a special type of cell. They are characterized as polarized epithelial cells. And sorry, this is a typo. They have dendritic ends, which receive signals, and they have axons, which transmit signals. So polarized epithelial cells assemble into sheets, and they absorb and secrete. That is the hallmark of an epithelial cell. Signaling happens on the apical surface of the neuronal cells where there are cilia and microvilli. Neurons are highly specialized. There's even distinctions. They're specialized like within the neuron as itself. So the distinctions between like the apical and their basal surface, and that is what allows them to transduce signals from their dendrites to their axons. So the most important thing is that polarized epithelial cells are special because of the fact that they can be polarized. So we can have that kind of special specialization of the dendritic end, which is receiving signals, and the axon end, which is transmitting those signals. So during neurogenesis, growth cones guide the development of our nervous system. Chemoreceptors on the growth cone of the axon guide the development of our neuronal connections in early development of our nervous system. The axon growth cone is a specialized structure which extends from the end of the axon. That is considered a highly motile part of the axon that grows by expanding a specific part of it called the lamellipodium. The lamellipodium is directed based on the antenna of the growth cone, which is the end of the lamellipodium, which is called the filopodia. They form and dissolve during expansion. The filopodia is formed by actin cytoskeleton, which lends to the motility of the growth cone. The goal of the growth cone is to follow chemical neurotrophic signals in order to form synaptic connections. That is the development of our nervous system. So these different moving parts that we have, the lamellipodium and the filopodia, at the end of the lamellipodium, at the end of the axons, we have the axon, and then we have the little growth cone at the end. That is going to respond to chemical signals around it in order to attach itself or create a network of neurons. The elongation of the axon sheath is due to a microtubule cytoskeleton. Actin binding proteins are found in the growth cone cytoplasm abundantly so that the growth cone can expand through the lamellipodium, and the tubular and microtubule runs parallel to axons so that the axon can have some structural integrity during that growing and changing process. So that was a lot of information. Over here, this is kind of showing like the developing axon, the developing neuron over time, and all of that growth is happening at the growth cone. So the growth cone is extremely important because when you're studying the growth cone, you're studying how our neural networks are built and how our neural networks are, like, where that connectivity is coming from. So we really, the reason that I went into mentioning like the different proteins that are involved, or like the different components of the neuron, of the growth cone, is because those are going to take a hit during these neurodegenerative disorders. That is where the neurodegeneration is happening. It's happening at the synaptic ends of these axons, which have which used to have very good structural integrity. That is being degraded, and usually the components that lend themselves, such as the tubulin for the microtubule cytoskeleton of the axon, and the actin that is available in the cytoplasm surrounding the growth cone, those are not necessarily depleted, but the axon is not going to be developing anymore. After a certain point, like we start aging, we don't really have new connections being formed. And neurodegeneration, neurodegenerative disorders, they're accelerating that natural aging and having significant impacts on the motor function and the cognition of the people who are facing these disorders. So what happens to the growth cone when you have a neurodegenerative disorder? This is basically kind of tying in everything that I talked about with neurogenesis in relation to neurodegenerative disorders. So cytoskeletal arrangement abnormalities might commonly play a role in neurodegeneration seen in conditions such as Alzheimer's disease, cerebral ischemia, and multiple sclerosis. So that cytoskeleton, that microtubule cytoskeleton, in people who have Alzheimer's disease, it's been found that they have certain irregularities that are causing knots, basically, in the cytoskeleton arrangement. The normal structure of the axon, the normal structure of that that is needed to be maintained, it's starting to get knotted and tied up, and it's starting to seem very irregular than it normally seems. In AD, tauopathy induces a range of irregularities causing neuronal degeneration, including disruptions in axonal transport and functions of the mitochondria and lysosomes, as well as other microtubule-associated functions. So again, specifically for Alzheimer's disease, and also kind of for multiple sclerosis, and also not in the exact same way, but for all of these diseases, to some extent, what I'm trying to convey here is that the structure of the axon is under threat. It's not the same as it used to be. It's not capable of what it used to be capable of for several different reasons. And restoring that structural integrity is there. The question is, if we do that, if we're able to repair the axon, are we going to be able to repair those connections? Are we going to be able to see a meaningful improvement in the quality of life for patients that have neurodegenerative disorders? Glial cells provide support to neurons through secretion of neurotrophic factors like the nerve growth factor. That's what guides the axon growth cone. Brain-derived neurotropic factor, so that's going to be another one of those chemical signals that are being followed. Neurotrophin 3, another chemical signal. And then GDNF, which is, these are just a host of the kinds of sense that the axon is following, that it's like the juice that's glial cells are like the juice that surrounds our nervous system, and they have like a bunch of first aid things that they can supply to our axons, and they also have all of these signals that they can provide for the growth and the changes in the axon. So these are interesting because we get to, if we are trying to focus on neurodegenerative disorders, we also have to pay attention to how our nerve cells are responding to, over time, these different chemical signals. Is there degradation there? Is there room for improvement there? To go back to this point, to these 2 points over here, irregularities are stemming from either excessive activation or insufficient inhibition of different kinases and phosphatases, which are different types of enzymes that are aiding in the reactions that are forming and helping grow these nerves. They govern the interactions of the microtubule associated proteins, which are going to help with the formation of the axon, and neurofilaments within the neuronal cytoskeleton that they are governed by. That is why it's really important here what they're talking about with the function of the mitochondria and the lysozymes, because if we don't have energy in that cell, if we're not supplying the energy, we're not going to be, it's because we don't have that activity in those kinases and in those phosphatases to carry on those reactions. So it is really unfortunate. Obviously, a lot of this is like related to our natural aging, but yeah, it's clear that when you have a neurodegenerative disorder, it's like genuinely tearing apart the fabric of your nervous system. And facing that obviously is going to come with a whole host of problems that people would not encounter if they were aging without having to go through a neurodegenerative disorder. Magic of mushrooms. So, I was able to find that there are quite a few researchers that have already, for many years, been looking at the capabilities of mushrooms, and the active components of some of those mushrooms have been found to increase the size of growth cones and aid in neurotrophic signaling required for the functions of the cytoskeleton. So all of these issues that I was talking about here, kind of on this side, we're talking about the issues in the growth cone. We're talking about the structural integrity of the axon. And then on this side, I mentioned the different neurotrophic factors that are supposed to be providing that support for the growth and the direction of the axon. All of these, in the event that you have a neurodegenerative disorder, are taking a hit. Mushrooms, the magic of these mushrooms, some of them have been able to not only increase the size of growth cones by having provided support for that cytoskeleton that we keep on mentioning, but they're also aiding in the neurotrophic signalings. They're also aiding in the reception that we have to NGF, the nerve growth factor, to BDNF, to things that, to the chemical signals that are guiding the direction of our neurons so that they can form connections with other neurons so that we can have a well-structured and organized nervous system. So I compiled here, the rest of this presentation is going to basically be a summary of these 4 articles that I found were very educational in talking about how specifically we're using mushrooms to tackle this problem. Obviously, the person where I am at in my life right now, I do have enough background knowledge to explain as much as I have so far. But if you're interested in knowing more, you definitely should take a look at these articles. They are very detailed. If you're a George Mason student, you can access these articles wherever you would like to on through our they're available on several of our research databases that we have access to through George Mason University. So please, if you're interested in any one of these 4 articles that I'm about to show you in this very quick lit review type presentation, then take a look at them. It's really interesting to see how we can use the natural world to treat a disorder that is occurring naturally within our body. I think that that's a really interesting and beautiful perspective. And obviously, like many of us have seen throughout the course of the semester, mushrooms have been used for hundreds of years for their health benefits. But if we can use the advent of Western medicine, if we can use that deductive reasoning that we have equipped ourselves with, that scientific method, we can really actually apply some of the work that these researchers are doing into the ways that we treat neurodegenerative disorders, hopefully someday. And it would be really cool to see more research in that. So the articles here, we have therapeutic applications of mushrooms and their biomolecules, along with blah, 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 blah. I'm not going to read the whole title. I'm actually just going to be including a little blurb and a citation for each of these. But so over here, this is talking about the general therapeutic applications of mushrooms for neurodegenerative diseases. This is talking about a specific mushroom and the treatment of Parkinson's using a mouse model. Love those. This one is Tinospora cordifolia. And then over here we have Phaumatosis officinalis, which is actually the mushroom that I chose for my mushroom of the day, I believe. And then we have this 4th one. I don't remember if it's in the title. Oh, right here. Hericium ericineus, I think. That's just a lion's mane mushroom, I believe. So you get to see how those are supporting some of that neurotrophic signaling stuff that I talked about earlier. If you take a look at that paper, which is actually available online for free, and you don't really need to go through any research database to read. So very fun, very exciting. It's also the oldest paper. That's why I put it at the end. That's too much information. I'll just move on. So first paper, therapeutic applications of mushrooms and their biomolecules. So neurodegenerative disorders, NDs, are characterized by progressive brain or nervous system deterioration. Recent research, as stated in this paper, explores alternative treatments with edible mushrooms showing promise. Compounds from mushrooms like Grifola frondosa may prevent diseases like Parkinson's and Alzheimer's. Medicinal mushrooms contain natural compounds with anti-cancer and antimicrobial properties. These compounds include polyphenols and polysaccharides that are being studied for their potential in treating neurodegenerative disorders. These polyphenols and polysaccharides, that's the structure of the neuron. That's what that's lending to. It's also lending to that how we were talking about how neurodegenerative diseases, they're impacting like not only the neuron, but they're also like hitting the mitochondria. They're also hitting the organelles. So being able to see that these compounds found in certain mushrooms in treating diseases like Parkinson's and Alzheimer's are able to kind of rescue almost the functions of these organelles. That is very interesting. And it also offers cost-effective alternatives for drug research against neurodegenerative disorders. So there's also an economic value, perhaps, in studying mushrooms and isolating their active components with the assumption that they might be available abundantly in the natural world. So that is a very interesting perspective introduced by this paper. Next, Tinospora cordifolia suppresses neuroinflammation in the Parkinsonian mouse model. Mouse models, this basically just means that we had a bunch of mice that have been genetically modified to have, to be more, I think, not to have Parkinson's, but to be susceptible to Parkinson's, or maybe they are genetically modified to actually have Parkinson's. And you're studying the impact on those mice specifically. And it's great to use, actually, I'm not going to go as far as saying it's great to use mice. Obviously, people have their issues with that. I'm just going to say that it's great to use mice for this case because we're able to get meaningful information from studying mice that can be used for human beings. So Parkinson's disease involves the progressive loss of the basal ganglia neurons. Oxidative stress and neuroinflammation are implicated in PD pathology. Tinospora cordifolia aqueous extract shows promise in mitigating PD symptoms in mouse models. So this over here, it's talking about activation of certain genes to produce it's reducing the activation of certain genes and the production of certain compounds that cause inflammation. And since it's reducing their activity, then it's reducing the inflammation. This over here, TNF alpha, this causes neuroinflammation. It also decreases markers of microglial and astroglial inflammation while restoring levels of pro-inflammatory cytokines. So again, we're getting back into rescuing some of the functions of the cell. And this is one of the more complicated papers, so I can only go as far as what I can explain. But basically, neuroinflammation is being treated possibly by the use of this mushroom, and its active components have been found to reduce the production of certain compounds which cause neuroinflammation, not only in the nerve itself, but also in the glia. It also is like microglia are present in the brain when you have some sort of disorder. They're there to check in on things and fix things. And so if you're seeing if you're seeing decreases in the presence of these markers, then that means that you're seeing better performance of your nervous system and also a reduction in that inflammation. So very cool stuff there. This is the Phamatopsis officinalis, which is the agarikon mushroom. Great mushroom, been used for hundreds of years in traditional medicine to treat a bunch of things. It's great for immunity, apparently. It's great for inflammation. And also it might be great for it might be great for the treatment of neurodegenerative disorders. Specifically, this paper is just talking about the general medicinal use of this mushroom, and how it is and how they extract the compounds from the agar-akan mushroom. But most importantly, I just want to kind of go over this right here. This is the part that relates the most to the neurodegenerative disorders. So again, they're able to rescue the function of the neuron cell organelles. They're able to provide the polysaccharides and the phenolic compounds that we need in order to restore the function of our nervous system, perhaps. So very cool stuff there. And also it's oh wow, not even hundreds of years, sorry, 5,000 years back. It's also like very endangered, sadly. And maybe by noting its importance, maybe by taking a look at how it can be used for so many things, including making the lives of people who are facing neurodegenerative disorders easier, that could be a cause for stakeholders to want to protect the wildlife and want to kind of step in and sustainably grow this mushroom so that we can use it to save people's lives. And then the last one is Hericium erinaceus, which is the lion's mane mushroom. This study investigates the effects of ethanol extracts from 4 edible mushrooms on nerve growth factor gene expression in astrocytoma cells. Often, or among them, Hericium erinaceus. Oh my goodness, I'm so sorry. Among them, the lion's mane showed concentration-dependent promotion of those neurotropic signals that I had mentioned earlier. So of the gene expression for the nerve growth factor, so that we can have more secretion of the nerve growth factor protein. This enhancement was mediated by the JNK pathway, as shown by the inhibition of this specific gene. It's going to talk a lot about experiments in mice again. And this part over here is really important. So the hippocampus, that's one of the sites in our brain that shows the longest, that is where we go through neurogenesis for the longest period of time, because we're developing memories for a long time. That's like the jiggly part of our brain. So if we're seeing that there is an increase in that nerve growth factor gene expression in the hippocampus, we're seeing that there is an enhancement to that neurogenesis, to the guidance of our nervous system and the formation of those connections in our brain, specifically within the hippocampus, through just the administration of a lion's mane powder. That is extremely interesting. Obviously, this is, like I said earlier, the oldest paper that I had taken a look at. This is 2008, so that's a while ago. But ongoing studies are still able to find that lion's mane mushrooms have a significant, not significant to humans yet at least, but have made significant impacts on the neurotrophic signaling within the nervous systems of mice in several studies. So that is something that could be very interesting to take a look at. And my conclusion, just studying these papers has made me kind of wonder about the implications that this might have. And honestly, I believe that using mushrooms to treat neurodegeneration is a possibility in modern medicine, and it would introduce new perspectives in both mycology and in Western medicine. The use of mushrooms goes back thousands of years, and it is very important to modern medicine because of the fact that it's like in the framework of how we develop like our scientific method, like I said. Like, if we're taking a look at the traditional, obviously, traditional medicine there are valid concerns that people have with traditional medicine, but there's no reason to completely abandon what people were using for thousands of years to feel better and not go in with our technology that we have today to find whether or not there is medicinal use of these mushrooms now today, in the world that we live in, especially if it can improve the quality of life for people who are facing these neurodegenerative disorders. So thank you so much for your time. Sorry if I sounded confusing. It is a confusing topic. I wanted to do my very best in presenting it. And I wanted to give it my best shot. It's something I'm really interested in as it relates to my major. And I honestly was able to learn a lot from creating this presentation as well. So if you were able to learn a lot, I'm glad. If you were able to learn anything at all, I hope that you learned that mushrooms are magical and and they can be used in millions of wonderful ways in medicine, possibly if we gave credit to them where credit was due. So thank you very much for your time. I really appreciated taking this class this semester, and a lot of my understanding of this topic was made possible due to this class. So have a great rest of your day, you guys.